Hello, my name is Brittany, and I'll be explaining the stimulation of gastric acid secretion by rabbit parietal cell A2B adenosine receptor by Rosa Maria Arin and colleagues. These findings were published October 14th, 2015. The main focus of the article is on this secretory pathway, the receptors, and the ligands that are also involved. This secretion that we're talking about happens in the parietal cell, so I think it is necessary to talk about what is known about the parietal cell hydrochloric acid production. The parietal cell's main focus is to secrete hydrochloric acid into the lumen of the stomach. These cells get the components necessary to form the HCl from the blood. The hydrochloric acid secretion is achieved by the movement of these tubular vesicles that form inside the cell, and they move toward the apical surface. And contained in the membrane here are many proton pumps that are necessary for the exchange of molecules in order to produce the hydrochloric acid. The tubular vesicles move along actin filaments in order to reach the apical surface. This movement in the ATPase channels that are on the tubular vesicles use ATP to fuel their mechanisms. The ATP comes from the many mitochondria that are found within the cell. Stimulation of this parietal cell to produce the hydrochloric acid is initiated when G cells release gastrin that activates the neighboring ECL cell to produce histamine. Histamine is released from the ECL cell, and it binds a G protein linked receptor found on the membrane of the parietal cell. The binding of the histamine causes a shape change, and the G protein is released. It then binds adenylate cyclase, and using ATP, cyclic AMP is produced as a second messenger that initiates a cascade of reactions that allows the production of the hydrochloric acid. So these channels allow the chloride ions to pass into the lumen of the stomach, and the ATP also powers the channel that exchanges the hydrogen ions for the potassium ions. At this point, the parietal cell has now passed hydrogen and chloride into the lumen, producing hydrochloric acid, so the stomach can begin its regular digestive functions. So withdrawal of the stimulus allows the tubular vesicles to collapse back down into the cytosol, inactivating the parietal cell. And in rabbits, adenosine is a known ligand that binds to a similar G-linked receptor, and it might be used in rabbit gastric secretion pathway, but they weren't sure. So this is the area in which the researchers of this article studied. The experiments are with rabbit gastric mucosa, and they are investigating adenosine and its receptors in relation to the secretory pathway. So adenosine is a nucleoside. It's like a metabolite that is continuously produced either intracellularly or at extracellular locations, and it modulates different physiological functions. The adenosine effects are mediated by, there's 4 different subtypes of the adenosine receptors, and they're all G-linked receptors. There's the A1, A1A, A2B, and A3. The A2B receptor is the most unknown of them because it has a relatively low affinity. So the A2B receptor is, of course, the receptor of interest in this case. These scientists know a little bit about the receptors that and they have performed some of the research on their own that has led to further insights into this investigation. But I'd like to briefly go over what they did know before they began this research. So they knew that there were high levels of the A2B receptor expression in different parts of the gastrointestinal tract, where it seems to be involved in activities such as vascular tone, inflammation, secretion, and motility. And there's little known about the relevance of the A2B receptor in the gastric physiology. It has also been reported that the adenosine A2 receptors are on parietal cells in rabbits that modulate gastric secretion, And pharmacological studies have shown that adenosine and its analogs stimulate gastric acid secretion in parietal cells from rabbit gastric mucosa. So the scientists of this research had already performed experiments demonstrating that the A2B gene is expressed in the mRNA in the rabbit gastric parietal cells, and they have confirmed its presence through flow cytometry. So this wealth of knowledge led them to their hypothesis. Arin and colleagues hypothesized that adenosine and its analogs might have a role in gastric acid secretion via the A2B receptor binding in the parietal cell. So they used parietal cells that they got from normal rabbit gastric mucosa. And figure 1 here is showing saturation curves of the kinetic behavior of H-NECA. H-NECA is a radio-labeled adenosine ligand, and binding assays were performed to determine the affinity, and using the data that they collected, they created these saturation curves that you see here. So in figure A, you can see that as the concentration of the H-NECA increased, so did the binding of the A2B receptor. Figure B is showing that an equilibrium occurs over time, and figure C is indicative of competitor assay that was performed for an hour. The drop that you see indicates the ligand coming unbound from the receptor. Also, due to time constraints, I won't be able to go over all the data they collected, but I'm going to give you the important conclusions, and if you are interested, I can answer any of your questions, or you can read the paper. So anyway, from these assays, they were looking at affinity values that indicated that the ligand binding is antagonized only by the A2B receptor agonist NECA. So like the NECA is the agonist. And also it's not shown in here, but they were able to determine that the A1 and A3 receptors were not found on the parietal cells, but I didn't include the graph for that. So anyway, the 2-CADU is also an agonist for the A2B receptors, and to analyze the involvement of the A2B in the process of the gastric acid secretion, they measured the aminopyrine accumulation to determine the acid production under the CADO increasing concentrations. So in these graphs, you can see that the increase of the secretion and their corresponding standard deviations. Here we are looking at the adenylate cyclase activity on the parietal membrane in the absence or the presence of the 2-CADO. And the adenylate cyclase activity was measured in cell membranes by quantifying the cyclic AMP that was generated from the ATP substrate. And this was done using competitive protein binding procedures. But once again, there's a lot more data that is not presented here, but the take-home message is that the adenylate cyclase, which is important in the secretion pathway, is stimulated in these experiments with the A2B receptor. So the scientists that published this article concluded that the A2B receptor is the only adenosine receptor present on the parietal cell surface of the 4 subtypes, and it is involved in the acid secretion pathway. So why is this important? This is important because these findings suggest that the A2B receptor can be targeted for drug treatment of tissue hypoxia to control inflammation. Thank you for listening.